Hello, everyone. I am Yu-Cheng CHEN from Nanotechnological University in Singapore. Today, I'm very pleasant to have this opportunity to present my work on biointelligent lasers for sensing and bioinformation encoding. Lasers. We are all familiar with lasers, which is a powerful tool in our modern lives, including medicine, industry, communication, and military. The scale of laser has also become smaller and smaller, from the very first laser in the 1960s to laser diodes in the last decades. Nowadays, lasers have become even smaller than a piece of hair, advancing to the micro to nano scale. Laser has the unique property of narrow line width, strong intensity, and high contractility. So what is actually a living laser? Compared to a conventional laser made of bulky crystals, living laser replaces the gain material inside with living biologicals, such as bacteria, DNA, proteins, and even cells. Such living biological materials could therefore respond to its surrounding environment and stimulus. Here, I will present some examples of how micro to nanoscale living lasers can be transformed with intelligent functions. A laser which can feel, sense, and think. Recently, we have developed and demonstrated tiny lasers which can respond to peptide folding at the biointerfacial layer. The middle one shows an example of a living laser printed on a chip formed by bacteria, which can respond to drug interactions. We also developed microlasers which can respond to protein conformation changes, especially to neurological disorders. The similar concept can also be applied to human cells and tissues. For example, tiny fiber lasers detecting cancer in human blood and human tissues were developed. Based on this concept, a new imaging modality called laser emission microscope was invented. Lasers could also be embedded inside cells for sensing and tracking its intracellular environment. Finally, tiny lasers can also be applied for information encoding and prediction. For instance, we use DNA and protein molecules to control the laser signal for information encoding. We also train the laser signals with artificial intelligence to predict the cell types in brains. Lastly, I envision that the future of micro to nanoscale lasers could be used as a stimulation light source for therapy. It could also be injected in human body for monitoring health conditions. It may also be implemented as a wearable sensors to perform smart functions in the future. Finally, I would like to thank all the committee members and thank you all for your attention today. Thank you. Goodbye.